Hello, beautiful viewers. This is Crew Diamond Clouds. Today we are extracting DNA from a banana. And as you can see, I have my banana. I'm chopping them off into smaller pieces and put them in a plastic bag. I have two groups of students for this experiment. I have the boys and the girls. I want to see who is going to make the best DNA extraction. So the first thing we did was the students put in the beaker, they pour 100 ml of water, and we're going to create a mixture of salt, water. So right now they are adding 2 spoons of salt to the water, and then they will stir it up. So we add salt to this experiment because the salt is going to help the DNA stick together. That's why we added the salt into the water. Yes. So that is why we added the salt. And then the students now mash the banana. You have to mash it so that when you add the mixture to it, it can mix properly. And now they are adding 10 ml of dish soap. Now this dish soap is very important in this experiment because it's going to break the cell membrane to release the DNA, because the DNA is in the nucleus and we want to break it out. Yes, the cell membrane is a barrier for the cell, so that's why we want to break that with dish soap. So that's why right now we got 2 things in here. In this mixture we have dish soap and we have salt in this mixture. The salt is to make the DNA stick together, and the dish soap is what? To break the cell membrane. So we want to break the cell membrane, release the DNA, and want the DNA to stick together. Now we pour the entire mixture into the mashed banana. Yes, that's what the kids are doing right now. They're gonna pour that mixture of salt and dish soap into the banana, the mashed banana. Yes, and after that, now they have to filter the whole thing. The mixture of salt, dish soap, water, and mashed banana. You have to filter it to remove all the impurities so that it can be clear before we start the extraction of the DNA. So now we're pouring the filtered mashed banana, dish soap, and salt into the graduated cylinder before we start our extraction. Now to do the extraction, we use very cold alcohol. I let this stay in the freezer, not actually the freezer, but in the fridge for some time, and then the students went and brought it back to the science lab. Now we're gonna pour 50 ml of that cold alcohol into our filtered mixture to extract the DNA. As you can see, the DNA is now coming on top of the whole mixture. The DNA is that fluffy white thing you see up there. And the reason why we added the cold alcohol is that the cold alcohol makes the DNA come out so that we can see it very clearly. It makes it stick together. Yes. So that's the DNA right there. Thank you for watching. Bye bye. Yeah. Watch out for more videos.